Hi, everybody. So I'm Neva Durand from the Aiden lab, and this is going to be a talk that's a little different than the ones you've seen so far. This is a basic research question that we're trying to address, but you should maybe get to learn something new, and we do use some deep learning in trying to figure out some new and interesting things about science. So the human genome is long. It's physically just a large molecule. It's 3 billion letters long. It's 2 meters long if you stretch it out from end to end. But somehow it folds up upon itself to fit into the nucleus of a cell, which is 6 microns wide. And so the question is, how does it do that? And the way in which it folds inside the cell actually is a really important question. It's a really old, open question, and it's one that actually has implications for human health. So how does it have implications for human health? Well, every cell in your body has the same DNA. So you look at your skin cells, you look at your blood cells, but they all look different under the microscope. Why is that? They're all expressing different genes. They all have different function. The way in which the DNA is folded inside the cell, the way that the cell packages its DNA, is crucial to maintaining its functional state. And it's also crucial for things when they go wrong. So cancer cells also have DNA, often that looks the same as the rest of your DNA. And as your body ages and it gets older and your cells start to do odd things, express or overexpress or underexpress genes, that's also something that's implicated in epigenetics and how the function of the cell is changing over time. So one of the ways in which this manifests itself is in the genome spooky action at a distance. So you can imagine a linear genome, like a viral genome. A viral genome is very simple. You have a promoter, it's next to a gene, and the promoter turns the gene on. It would be like if you had a television set, you just push a button on that television set. But nowadays, things have advanced, and so have genomes. So mammalian genomes, like the human genome, have things called enhancers in them. And the enhancers turn up or turn down a gene's expression. And the enhancers are far away in the linear genome from the gene that they affect. And so there's a question, this makes it actually really difficult to figure out what's happening. It makes it difficult to analyze because you can't just look linearly next to what you're looking at. You need to try to figure out where these enhancer elements are. And one way that people think this is happening is that the enhancers are brought into close proximity with the gene via folding. And so the question is, how can we find these loops? How can we find these places where the genome is touching each other in 3D when maybe it's not close together in one dimension? So that's the problem that we look at. We look at this problem of mapping the loops. Now, when you want to try to figure out where something is, normally you would try to look at it. But it's actually a very difficult problem to look at things in the cell. They're very small. In microscopes, you're going to have to do 3 dimensions to actually figure out where you're looking at. You use these techniques that actually don't scale that well and are sort of difficult to do genome-wide. So there's other ways to look at structure from data, and one of the ways to do it would be like, say, the way Facebook does it. If you were to try to figure out something about the structure of relationships from, for example, Facebook data. So let's look at an example. This guy is Homer Simpson. You might have heard of him. He lives in the fictional town of Springfield. And maybe we're curious about knowing things about the relationships in Springfield. So we might look at Homer's Facebook photos, and he has a picture of himself with a donut. So it's something that he likes to do a lot. There's pictures of him with this other individual. We see also some pictures with more individuals. He seems to be frequently with them as well. And then there's some other people. And we can fill out this whole slide with pictures from Homer's Facebook. And we could take that information and try to figure out something about the structure of the Springfieldian relationships. And in this case, what we see right away, if we make something called a contact map, and we just take every time we see Homer or anybody in a picture with each other, we just increment this map. So it's like a heat map of when people are in contact with each other. And what we see is that immediately structure jumps out at us. We can immediately see this square in the middle. That's Homer's family. And we can even see that there's an enhanced area of contact at the corner, and that's the relationship of the marriage between Homer and Marge and how they've nucleated this whole family of things in between them. So we want to do the same thing, but for the genome. So what we're going to try and do is take pictures like Facebook pictures, but for the genome. And we do this via an experiment called Hi-C. Hi-C is an experiment that was invented by my PI, Aiden. I'm part of Aiden Lab, and I've been working on this kind of data now for about 7 years. And I'm just going to quickly tell you about the experiment. Basically, what we do is within the nucleus of a cell, we're looking at the DNA. We cross-link the DNA, which freezes it into place with formaldehyde. We cut it with a restriction enzyme, which is the type of genomic scissors. We fill the ends in and mark them with biotin. And that's going to tell us where we want to look. So that's going to give us like an idea of this photo of that 3D interaction. Then we ligate, which is like a glue that glues the DNA together. We purify and shear the DNA. We pull down the biotin, and then we sequence using paired end sequencers. And we end up with massive amounts of data. And from that data, we're going to create contact maps, the same kind of contact maps you just saw with the Homer-Simpson's family, but on the genome. So on one axis, instead of the pictures, we have the genome. And on the other axis, we have the genome. And right away, you see some structure. You see that there are boxes along the diagonal. And those boxes, how many boxes are there? There are 24 boxes, or 23 boxes. Why are there 23 boxes? Because there's 23 chromosomes. So this is because we know chromosomes actually like to hang out with each other. They're in these things called nuclear territories. And so if you're on chromosome 2, you're not really interested in hanging out with chromosome 19. And that's why we don't see a whole lot of chromosome 19 interaction with chromosome 2. And we do see a lot of things here. And this has been posited for a long time, since the 19th century. It's known from microscopy, but it's a nice positive control for the experiment. So we can zoom on in to chromosome 8. And again, what you see right away is that there's like lots of interesting things happening. In particular, there's lots of interesting things happening off diagonal. Now, on diagonal, that's that big red thing. That's probably the first thing you really notice. That is just the fact that things are close together in one dimension means that they're going to be close together in 3 dimensions. That's just physics. But then also you do see a lot of structure off diagonal, and there's lots of really interesting things happening. And I could tell you about many, many of them, but I'm going to focus on one thing. And also, we often get a question that I want to answer right away. The square in the middle is the centromere, so there's no mapping data there. So we were interested in looking at loops. What does a loop look like in the contact map? Well, you have this red dot. If we're interested in this loop that's A and B together, then you've got A is in orange, B is in blue, and the things that are next to A are red through yellow. The things that are next to blue are essentially lighter blue through pink. So we can make a contact map that's going to look like that. And we can try and imagine what that's going to look like. It's going to look like a lot of contacts between A and B. And then it's going to kind of diminish as we go away from there. So what we see are these. We see these loops become these things that look like focal peaks in the contact map. And so we were able to find about 10,000 loops. This is what they look like. You might have this strong diagonal, but the diagonal here is 193 contacts. On this loop that's off diagonal, it's 281. So it's quite intense, especially considering that there's a bunch of stuff in between that's not nearly as intense. And it's the same thing down here on chromosome 13. We did this. It's a biological experiment, so we have replicates. We can reproduce this data. And we can also actually verify it via 3D FISH. So in this example, we have a probe on a putative loop, which is the green and the blue. That's the like blue line you see. And the other blue, the red arc you see is the control. And when we look under the microscope, we see that when we put probes there, fluorescent probes there, we can look under the microscope and we can see that the green and the red are in contact, as we had predicted, and the red and the purple are not in contact. So what does this have to do with genetics? As I told you before, what does it have to do with enhancers and promoters? Well, we have this nice example of something that we saw in our data, which is that those loops can turn the genes on and off. So on the top 2 lines here, you have activation marks that are telling you that this gene is active. In this case, it's this gene Adam TS1, which is known to be expressed in the cell line IMR90, but it's not expressed in GM12878. And as you can see, the fact that this gene is expressed seems to have brought in these things into contact with each other, these promoter enhancer loops, and has really changed the structure, the 3D structure that we see in the data. So that in turn has this, what I was saying, these implications for the different kind of cells. So how do we do this? What do we do with these contact maps? How do we find these features? It turns out that finding these loops can be really difficult, can be a really difficult task. prior to sort of the main focus of the work that I had done, these were the kind of loop calls that people were making. And probably, in one case, it's a different experiment, so they didn't quite have, there's probably some biases in the experiment that weren't taken part of. But in the other case, it's probably just that they had some kind of expected model that had to do with distance apart and being enhanced over distance apart, for example, or enhanced over genome-wide average. But this was not actually giving very good results. In particular, it was finding areas of maybe enriched contact, but not of this focal enrichment that we're really looking for when we're looking at promoter enhancer loops. So we were able to find areas of enriched contact, more focal loops. The algorithm that we used to do this was called hiccups, and it's basically like a filtering algorithm. So it just goes across the whole area. It looks in this window, and it looks and sees all these filters, and actually makes sure that this areas of enrichment is over any of the filtered areas. Now we're doing a convolutional neural network for this. We're doing something very simple right now. We've just taken something off the shelf and applied it to our data. So what we're doing is taking our data, and in this case it's just a chromosome heat map at a certain resolution, in this case at binned at 5 kilobase resolution, chopped it up into 15 by 15 tiles, and then we do convolution, pooling, flattening, et cetera. We do a couple layers of that, and then we get an answer as to whether or not we think that there's a loop there. And so our preliminary results are good. The thing that's most interesting to me is that we're able to find things that maybe we couldn't see before with our other algorithm, and even in the data itself, like as a human. So on the side here, we have here. This is the map that we're training on. It's like 5 billion read pairs. This is actually a deeper version of the same map. It's 42 billion read pairs, so it's a lot deeper. And basically, these are the loop calls that we get, and of course, they're also in the other data set. So this is with our previous algorithm. Now with our new algorithm trained on this data set, so still just trained on 5 billion read pairs, we're able to see these things. They don't actually look like much in that map, even to the human eye. But if we look in the deep network, you can actually see that there is signal there. These actually were loops. And this is a case that we see kind of over and over again. We're finding these things that we were like, oh, well, are these false positives because they don't line up with our previous loop calls? And we're like, actually, I'm not sure that they are false positives. We're going to need to go through and like manually label these and make sure that we're actually doing the correct thing. So that was the pretty exciting result from our point of view. So in conclusion, I just wanted to say gene expression is an interesting problem from a health perspective. It really is a very basic problem, but it really does have some health implications. And how DNA folds affects the gene expression. So using this deep learning, we can map the loops genome-wide. We're hoping to be able to do even more, not just the loops, but some of the other really interesting features, such as the domains that are along the diagonal. There's things called compartments and subcompartments. And we're hoping to be able to deepen our understanding of nuclear architecture and thereby hopefully add to like basic understanding of biology. And with that, just want to thank my lab. This is, I'm in the Aiden lab. That's Era's Aiden. This is all the people in the lab. They all do a lot of great work. All right, thanks. I'll take questions.